Hi all, hope you're ready to jump into this week's assignment. I decided to avoid a little bit of confusion that perhaps a video explanation of the assignment would be a little bit better than just writing an email or to follow along with the data in lab archives. And of course this week we're going to talk about what's in a color. Since we aren't able to do all of our own Drosophila matings and actually count our own results from F1 and F2 crosses, I thought it would be appropriate at this point in time to actually go ahead and do a little bit of an experiment. You've now seen the results of F1 and F2 crosses, both in lecture but also in lab. You've analyzed the results of the F2 utilizing chi-square analysis from the corn kernel counting experiments. And now we want to actually see what happens with something that has never been reported before. This is something we've only done here at Stockton a couple of times, and mostly on the basis of a research project done by, yes, undergraduates just like you. So, of course, we know that Drosophila are a very, very popular model organism in genetics. You know the background of Thomas Hunt Morgan and his discovery of the inheritance of white eye color as a sex-linked gene. And in fact, the first gene that was shown to be associated with a particular chromosome and led to the entire discipline of linkage analysis and ultimately genetic mapping, the culmination of which, of course, is our current understanding of the human genetic map. So really some very exciting stuff with very, very simple beginnings. Please remember, as you may recall from your lecture experience, that really it was Thomas Hunt Morgan's student, Alfred Sturtevant, who created the first genetic map using crosses among Drosophila, specifically sex-linked genes on the X chromosome in Drosophila. And at the time that he created the first genetic map, he was an undergraduate research assistant of Thomas Hunt Morgan, and eventually led to the entire field that we use routinely in our work, in whether you're dealing with animal systems, basic research, or in human medicine itself. So really exciting stuff, and started by an undergraduate. So never underestimate yourself. The two pigment systems that we now know determine the eye color of Drosophila are the pteridine and the Omechrome biochemical pathways. Now, you may not be familiar with the nomenclature, but let's go ahead and show you what it actually looks like. So on the upper path, you could see the pteridine system above. It functionally creates all of the red colored pigments that we see in the Drosophila eye, and the other is located below, which is the Omechrome pathway. The Omechrome pathway is based on tryptophan metabolism, the amino acid tryptophan, of course, and creates all of the brown pigment. So what this tells you is that in the wild type condition of Drosophila has both brown and red pigments, Omechrome and pteridine pathways respectively, okay, have both brown and red pigments, creating the normal brick red color that we see in Drosophila. Now in the pink ovals, you'll notice that there are indicated 5 known mutations in Drosophila that result in different eye color. If the pteridine pathway is cut off at the level of GTP, this is actually a GTP mutant, by eliminating the entire right-hand set of pathways, What happens is the only color that can be expressed is from the omechrome or tryptophan metabolism pathway, and so we get brown-eyed flies. There's another mutation a little bit further downstream which creates a sepia. It's called a sepia mutation, and basically it gets rid of the red, or the most prominent red pigment, which is called Drosophila, leaving a couple of yellow and bluish type pigments in the eye, and that in combination with brown creates a sepia tone color, or kind of a tannish, old-fashioned brown. You probably have a sepia filter on your smartphone camera that makes an old-fashioned looking like photograph. Alternatively, if we eliminate the brown pathway, meaning the tryptophan metabolism pathway, we can see a couple of different colors that result in only the reds being produced. And these both have very bright red names, vermilion and scarlet, of course. And these, by eliminating that xanthometin pathway at the end, eliminates the brown pigment, and you get these vibrant red colors in the Drosophila eye. So we can manipulate the pathways to create the kind of colors that we're interested in, and this was one of my undergraduate research assistants about 3 years ago had this great idea to manipulate the colors to create different eye colors of Drosophila utilizing combinations of these mutants. So the ones we landed upon were brown and scarlet. Really simple approach. Now, on the top right portion, you can see kind of the brick red view. These are all female Drosophila here, and you could see the brick red in the top right Drosophila. That is the wild type. It is dominant for both brown and scarlet, meaning both brown and red pigments are produced, and you get this kind of brick red color. Immediately to her right, our left, if you look at the left, you see the bright red eye. This individual is homozygous for the scarlet mutation, eliminating the brown pigment and creating this vibrant red colored eye. And the Drosophila on the bottom of the photograph is, of course, the brown mutation, which eliminates all the red pigments, the entire series of red pigments, and we end up with that brown color. So these are the wild type, scarlet, and brown mutations. It's a little difficult to keep straight in your head, at least in our heads as well, is that the scarlet mutation is a mutation in the brown pathway, and the brown mutation, sorry, reverse camera angle, the brown mutation is a mutation in the scarlet pathway, eliminating the red. Okay, so we have that kind of clear in our heads. So the thought was, what happens if we eliminate both brown and scarlet? And if you recall from looking at our combination here, our hypothesis is indicated in the middle section. If we eliminate both brown and we eliminate both scarlet, we should get a white-eyed fly. A white-eyed fly. Now, in fact, we do. And here's what our first set of white-eyed flies look like. You can see they have a very clear, non-pigmented eye structure. This white, okay, is a very different white than that identified by Thomas Hunt Morgan. Remember that Thomas Hunt Morgan's view of the white eye, let me straighten the camera a little bit. Thomas Hunt Morgan's white eye was sex-linked. This individual right here is not sex-linked, but is actually the result of a double recessive. That is, it is recessive small bw, small bw, small st, small st. So it is a brown, scarlet, double recessive mutation, eliminating both pigment pathways. All right? There is a really good thought if you're thinking about pigments and you're thinking, well, what was Thomas Hunt Morgan's mutation? And it turns out that the white-eyed flies that are sex-linked are actually not in either of these pigment pathways, but rather a mutation in a protein gene that codes for a protein that is a transport protein. And it is a protein that transports pigment into the eye itself. So a very, very different way of getting a white eye. You can get a white eye by eliminating pigment, or you could get a white eye by stopping the pigment from being transported into the eye itself. So really fun stuff. So you cannot do the crosses yourself. So what I've done is provided you with a spreadsheet. And in the spreadsheet are a series of different hatches and different vials within a hatch indicating the wild type, the number of wild types, meaning brick red eyes, brown, meaning brown-eyed, scarlet-eyed, and of course, white-eyed individuals. So these are our phenotypic categories. And notice we have a fairly substantial number of hatches and vials and days within those vials that we can count for you, and actually went through and counted the flies. So you don't have to actually do that portion of the experiment. Too bad, because it's a lot of fun. So what your assignment is first off, define your expectations. What do you expect to see? What we did was we took the brown and the scarlet mutants, crossed them. So our F1 generation is heterozygous at both the brown and the scarlet locus. Okay? Heterozygous. We then created an F2 population by intercrossing our F1 generation individuals. Our F2, you should know by now, this is a standard F2 dihybrid cross. So, what then would your expectations be to observe in terms of the numbers within each phenotypic category? What's your expectation? This should be automatic for you right now. F2, dihybrid cross. What do you expect in terms of phenotypic ratio? I'm not telling. So, you want to define what your expectation is. Then you need to summarize the data. Notice that the table follows the same general format as the table we used to count the corn kernels. And at this point in time, you can see why we counted each individual row of kernels, because each individual row of kernels is basically like a single vial of Drosophila. Something you should be thinking about, and should have thought about when you were counting the kernels, is looking at the ratios that you observed in each row of kernels. Was each row identical to the expectation as predicted by Mendel's theory? Probably not. So a question you have to ask yourself is, why not? And yet, when we summed all of the kernels in all of the rows, in most cases we approached the expectation. So our results approached the expectation as predicted by our theory and by our Punnett squares. So this is something you should be looking at as you look at the data in this spreadsheet. Okay, you should be thinking about this. Meanwhile, you can calculate your observed ratios. There's a little bit of confusion with people calculating ratios. Ratios are not just an arbitrary placement of the sum total of observed phenotypes within any particular category. Remember that a ratio is always expressed with regard to the lowest numerical value of a particular category. So if you have, for example, 27 to 9 to 9 to 3, you don't say 27993. You reduce it to 9 to 3 to 3 to 1. How do you get that? You divide by the lowest numerical category. So in the example I just gave, the 3 would be the lowest numerical category. You divide everything by 3, and you get 9 to 3 to 3 to 1. Same effort here. Most of you did that when you executed your observed ratios for the corn kernel data, but some of you have to remember to do it, and in this case, it will be required. Ultimately, what do you see? How do your observed ratios correspond to your theoretical expectations? Close? Not close? Similar? Not similar? What do you actually observe when you look at the data? Ultimately, observations are great. Looking at data is important, getting a sense of what you think is going on. However, ultimately, it is statistical analysis in the scientific method that allows us to confirm or reject hypotheses. So in this case, you will perform a chi-square analysis on the data. You can use the spreadsheet that was available to you in the corn kernel data. And in fact, there is space at the bottom of the spreadsheet for this exercise for you to create your own chi-square table. And upon doing it, what did you find out? Were any deviations that you saw with regard to your observed data? Did they conform to your theoretical expectation? Were any differences due to chance? Or is there something different going on, some biological phenomenon? What did you find out? What happened? If the deviation is not due to chance, then you have to ask yourself, why? What's the explanation? What's the point? And that will be the entire sum of the project. Pretty easy to execute. It is due by next week's laboratory. So take your time, think about it. You can replay this if you have questions. And I do believe there will be in this particular piece of software, they will actually be some written notes as well. I'll provide the link for that. All right. Hope you guys are staying well. Stay safe out there. Be careful. Don't forget, mask up. Take care, all.